Okay, so this is the last part of cellular biology. So we've got cell cycle. You'll probably have a question on this in the exam. It's quite a big part of this unit. Okay, so basically there's two things in the cell cycle that are important to know. So basically you're going to need to know what the cell cycle is. But knowing basically mitosis and DNA replication are the two main things in this kind of topic. Okay, so first I'm going to go through DNA and the general structure of the DNA. Then I'm going to go through into DNA replication, how that works, the enzymes involved, and then finally go into a bit of mitosis. Okay, so DNA stands for deoxyribonucleic acid. Okay, so from this name we can see it is a nucleic acid, okay, and it has a sugar in it, and that sugar is deoxyribose sugar. Okay, so I'll draw that out for us. So basically the structure of DNA looks like this. Okay, so we've got a phosphate, we've got a sugar. In this case, it is deoxyribose sugar. Okay, and then we've got a base here. Okay, so this whole thing, this is what DNA is made up of. This is one unit of DNA, and that is called a nucleotide. Okay, so DNA is basically made up of repeating nucleotides, one after the other, on both sides. Okay, and they're joined together through the backbone, so basically you'll have another phosphate here, and then another sugar, and then another base, and then another phosphate, then another sugar, then another base. Okay, and the same on the other side, base, sugar, phosphate, phosphate, base, phosphate, sugar, base, phosphate, sugar. Okay, so it's basically, it's just running parallel. So DNA, one strand, two strands of DNA make up DNA, basically. Okay, so let's talk a little bit about these bases. So in DNA you have 4 bases. A, T, G, C. We've got something we call a base pairing rule. Okay, so see that? Basically, the base pairing rule means that A always bonds with T, G always bonds with C. Okay, so far we've got our DNA molecule. Okay, so DNA is made up of repeating sequences of nucleotides. Each of these nucleotides has a base on it. That base can be A, T, G, or C. And each of these bases will always have a pair next to it because DNA is double-stranded. So that pair will either be A with T and G with C. Okay, the reason why A and T only bond together is because there's 2 hydrogen bonds between A and T and 3 between G and C. So G has 3 kind of bond places, so therefore an A can't bond with G because A has 2 and G has 3. It doesn't work. Alrighty, so that's basically all you need to know about the structure of the DNA. So double-stranded, bases in the middle, joined with hydrogen bonds. The phosphate sugar repeating units, we call that the DNA backbone. Okay, and basically, yeah, that's the thing that kind of holds the bases in the right structure. So the bases is what carries the information. Okay, so I need more paper. Alright, so the order of those bases, so let's say we've got A, T, G, C, C, T, A, G. Okay, the order of these bases along the DNA strand will tell will kind of, that makes up the instructions. It's like the instruction booklet for the cell. Okay, so let's say that's the gene for, I don't know, hair color. Okay, so if you've got black hair, if you've got brown hair, okay, a slight difference in this gene, so maybe a C is swapped for a G, okay, that'll tell you what hair color you have, basically. Okay, so that's how DNA is kind of made up. Alright, so now that we know a little bit about DNA, let's talk about DNA replication. Okay, so because DNA is double-stranded, okay, we're going to draw it like this. This is our DNA. Okay, so DNA is double-stranded. Alright, so each strand of DNA always has kind of an end. Okay, so it can either be a 5' end, which we do with a 5 and a dash, or a 3' end, which we do with a 3 and a dash. Okay, and in DNA, each of these strands, they run parallel to each other, but we call that anti-parallel. Because each side of the DNA strand, so let's say that's the 5' strand, and that's the 3' end, okay, it's going to be the opposite on the other side. So that'll be the 3', that'll be the 5'. Okay, so that's going to be important just a little bit later. Okay, so every DNA strand has an end. Okay, and it just depends on what end of which phosphate is hanging off the end. But don't worry about that. Just think about it as one end's 5', one end's 3'. Okay, so here's our DNA strand. So you got your bases, A, T, C, G, whatever, T. Make sure it's base pairing. Okay, it's a G. Okay, all along here. Now, because we want to replicate this DNA, we want to make 2 strands out of 1. And because there's base pairing, what we can do is we can split it down the middle. And see, if you split this down the middle, got A, T, C, G on this side. Doing the same thing over here, A, T, G, T, A, G, C on this side. Okay, so all I've done is put that one there and that one there. Okay, and what I can do then is, because I know that A always binds with T, C always bonds with G, I can do the same thing over here. Okay, and I end up with 2 identical copies of my first thing that I had. Okay, but you'll notice that this side here, which was this side here, that's come from the original strand, so we call that old. Okay, the stuff that I've just brought in to bond with, to make up the DNA strand, we call this new. Okay, so this is what we call semi-conservative. Semi-conservative replication. This means that half is new, half is old. Okay, so basically we need to go on to how did we get this. So what happens is you get a... Okay, so what happens is you got your DNA molecule. Okay, so now the cell's decided, oh, I need to divide. Okay, so an enzyme comes in called helicase. Oh, sorry, hold on. No, we need to start with the polymerase. Okay, so polymerase, an enzyme called polymerase, comes in, and basically what it does is it untwists the DNA, because DNA is actually in a structure that we call the double helix. So you've probably seen that before for DNA. So it just kind of naturally goes into this double helix shape. So what happens is the polymerase comes along and it kind of unwinds it, okay, so that it looks like this. Okay, then what happens is another enzyme called helicase comes along, and he's like the unzipper. He comes along and he unzips. So he moves down the DNA, kind of breaking up those hydrogen bonds, unzipping the DNA, so that you end up with free, so these bits are separated from each other now. Okay, and that allows this guy, polymerase, to come in with nucleotides attached to him. Okay, so here's our nucleotide, sugar, phosphate, base. Okay, and he jumps in and he brings them, so say this is a, what have we got first? A T. Okay, so here's an A, here's the T, that's now alone because helicase has come along and split it away from its partner. Okay, so now a new nucleotide comes along, polymerase brings that along, and it joins to the T. And so this would be that guy there. Okay, so polymerase is kind of like the glue, like the sticky, it's kind of the thing that comes along and it sticks all of these nucleotides onto the old strands that are now exposed. Okay, and it'll actually help, and it'll put in those hydrogen bonds and basically form the new strand. Okay, so the two enzymes that are important are the helicase, which is the unzipper, and the polymerase, which is like the glue. Okay, and that is DNA polymerase. You'll find it, we'll talk about RNA polymerase later. So helicase, polymerase. So these are two enzymes. Enzymes are super sensitive, so we'll talk about that in the next one. Okay, so basically, yeah, so the polymerase comes along, binds it back up again, and, you know, then that forms a new DNA. So then helicase can come along and replicate that and do it again, do it again, do it again. Okay, so the helicase and the polymerase, they just keep being reused. Okay, so the DNA can replicate all of its DNA. So replicate basically just means make two. Okay, replicate, make more of the thing. So why do we need to make more of stuff? Because cells need to divide, and for cells to divide, they need the right instructions. Without the right instructions, the cell's not going to do its job properly, and basically, cell won't work. Okay, so if you think about it, your skin cells are constantly falling off. You need new cells, otherwise you'd rub your hands to like the bone, you know, it's just not great. Okay, so you need new cells, therefore you need to replicate your DNA. So now I'm going to stop the thing and I'm going to go and get some more paper, and then I'm going to talk about a little bit about mitosis and then the importance of enzymes.